Assalamu alaikum and welcome back to another video. In this video, we are going to talk about microfilaments. So basically, if we talk about the components of the cytoskeleton, there are three components. There is the first one, microfilaments, the second one, that is the microtubules, and the third one, that is the intermediate filaments. So when we talk about cytoskeleton, we say that so the major function cytoskeletal elements they carry out in a body, that is to maintain the cell shape, they perform the cell division, all of the events which you saw in the cell cycle, that is the mitosis and meiosis, the formation of spindle, everything which is happening in the cell. In fact, if we talk about the endocytosis, if we talk about how organelle shape is maintained inside a cell, if we say how an organelle that is showing movement in a cell, all of this is done by the cytoskeleton, and that is what we are going to discuss. So there are three types of the there are three major types or components of the cytoskeleton, which is right in front of you: microfilaments, microtubules, and intermediate filaments. So we'll go one by one. First of all, in this video we are going to discuss about the microfilaments. You can see from this from the structure here on your slide, the microfilaments they have a kind of a filament like structure in which different monomeric units these are joined together, and they have a diameter of 7 to 9 nanometers. Similarly, you have microtubules. Now these microtubules these are similar to the hollow cylindrical cylinder like structure in which you will see the alpha and beta tubulin dimers. These are arranged on one top of another and they form this cylindrical pattern. Similarly, we have the intermediate filaments, and these intermediate filaments, these are 10 nanometers in diameter. Again, you will find a coiled coil-like structure, which again maintains the different shape and the structural support within the cell. Now, let's understand the dynamics of the actin filaments within our body. So, how does this process happen that all of these, since we said that these actin filaments, all of it, so this whole arrangement, it starts from an actin molecule. Now this here, right here, this one monomeric unit, that is a G actin. And now all these G actins, what they do is one after another, they are adding on to the monomeric subunits, and in a way they are forming a filament-like structure, which is basically performing the different kinds of functions which we see. Now the process it begins by simply these monomeric units, they start a process by the step of nucleation. Okay. So nucleation, it prepares the cell, basically, it prepares these monomeric subunits to join to one another and then proceed to the another step, that is the elongation. So nucleation is done. That means it is the first and the foremost crucial step where we simply say that it is a building block. So it's kind of a building block of, where afterwards you will say that the actin filaments, these are actually formed. Okay. So after nucleation, now since few subunits these are joined here, and now next it proceeds with this step of elongation. On both sides you can see that now the F actin, the G actins, they are now these monomeric G actins, these are joined one after another, and they form an F actin. Similarly, the process it goes on until it reaches to a steady state. Now, at the steady state, it is when you can see now there is a polarity in the actin filaments. Now, as the G actins they go on adding on to the plus end, on the minus end you will see that there is the depolymerization. That means each G actin it is liberated from the negative end. Okay, so this is called as the steady state. This is how the process happens. Now let's understand. So this is a graphical representation of the dynamics of actin filaments. You have three steps. First and foremost, that is the nucleation. Obviously, it takes certain amount of time to gather all of these G actins together. This is how it is represented. So this is the initial lag period. This is referred to as the nucleation. Then happens the next phase, that is the elongation, during which it starts the joining of the G actins, and finally until it reaches to a steady state where it's constant. So the amount of the actins which join together, the same amount it's liberated from the F actin filament. Okay. Similarly, you can see this one. This shows us the time graph when we have added. So if in under the in vitro conditions, what we do is if we put some amount of the G actins, So that means if we are adding this structure, so what happens is now the process it starts from the another phase. So that means now the process it so this step is done, and now the process starts from the elongation that is able that we can see from this figure here. So when we add the nuclei at a time, say at time 0, so it starts from the elongation and then again the steady state is reached. So that's the dynamics of the actin filaments. Now let's try to understand what are the different kinds of the proteins that are involved in the regulation of filament turnover by the actin binding proteins. Now, so we talked about these actins. So these G actins, these are the individual units here. The globular actins, which are joined one after another, and they form this kind of a filament-like structure. Now, how this whole process is regulated? This is regulated by certain proteins, by the actin binding proteins. Like first and foremost, we have a profilin. Now in the profilin, so profilin first and foremost, the important thing is that it helps or it aids in the polymerization. Okay, so this is what we see here. So on the positive end here, this on the positive end, this profilin it adds and it gets added in a GTP bound state. Okay, you can see in a in an ATP bound state. So when you have a profilin and it's bound to the ATP, it will help in the binding of the G actins to the plus end. Similarly, we have another molecule, we have another protein, the actin binding protein that is called as a cofilin. And now it has an opposite role as that of the profilin. Now what cofilin does is that it basically liberates G actins from the negative end. Similar to profilin but opposite role. And now when it is liberating it's liberating it in the ADP bound form. So you can see this one here. So this D form here, which is liberated from the negative end by cofilin, that is in the ADP bound state. Okay. Again, there is another one that is called as a thymosin beta 4. Now this is also important. This again, thymosin, it performs the function similar to that of the profilin. Now let's see what it does. So this thymosin, so you can see here, now we have this actin in the form in the ATP bound form. Now this thymosin, what it does it sequesters basically. So it sequesters this protein which is basically bound to the ATP. Okay. And again, if it makes it available, it will again join on the plus end. So that means profilin, it has a positive role. It helps in the polymerization, whereas cofilin, again, it depolymerizes on the minus end. Thymosin, it sequesters. That means also, so the kind of a cofilin and thymosin, they have the similar roles. Only the profilin that is involved or that is helping that is helping and promoting the polymerization at the plus end. So this is what is happening here. The same thing which we mentioned earlier though. So in case of the F actins, we have the polarities there. We have a plus end and a minus end. There are some other proteins, actin binding proteins, but they have different roles to play. Suppose we have the plus end, there is a protein called as an cap Z protein which binds on the plus end. Similarly, we have another protein on the negative end, that is the tropomodulin. Now the function of these two proteins is that they basically help it to stabilize. So they help, initially if we talk about the cap Z proteins, so this cap Z, it helps the positive end to stabilize. So this is the function of different actin binding proteins. Especially important is the profilin, thymosin, and cofilin. So now this is the regulation of the formin. We talked about formin and we said it has a crucial role to play because it goes on adding different monomers, the G actins on the plus end. Now, if we talk about the same, how the regulation of the formin is governed by the intramolecular interaction. Now see, this is a plasma membrane, the exterior of the cell, and this is the cytosol. Now all of the signaling which is happening, that is inside the cytosol, within the cytosol. Now let's concentrate on the structure of the formin. So this is a formin protein. Now when this formin protein, it's in the inactive state, it kind of a refolds upon itself. Now you will see in this structure, in the structure of formin protein, it has three domains. It has one that is RBD. It stands for the Rho binding domains. Similarly, it has an FH1 and FH2 domain. Now, but when it is inactive, it will refold on itself. But when there is a signal which basically binds and it activates, so there is a row in the form of a GTP, that means in an active state. Now, under such conditions, now the formin protein, it becomes activated. You can see this RBD, it's bound to the row. Okay, so this domain, it helps it to bind to this monomeric protein, the row GTP. And then this activation, it promotes the exposure of the FH2, while from the nucleation will start, and you will see that from the plus end it goes on polymerizing, and from the negative end it's depolymerizing. So this is how the profilin basically governs, or it helps. So there is a signal coming, and then we have the activation of the row GTP. Once the row GTP is activated, since this formin, it was initially, it's present within the cytosol, but it is present in the inactive state. So as soon as there is a signal, this row GTP, it activates, binds to RBD, activates it, this exposure of FH2, nucleates this, it begins the nucleation step. It will then elongate and then reach to the steady state. So this is how the this formin, it governs the formation of the actin filaments. Now after that, let's summarize all of the signal induced changes in the actin cytoskeleton. Earlier as well, we mentioned that it all starts from a growth factor. Okay, so there has to be a signal. Now this signal, it can be a growth factor or it can be a lysophosphatidic acid LPA. As soon as it comes, this signal, the ligand which we say, it will bind to its receptor. The receptor which is present on the membrane, and now this receptor, it is now in contact, or it's now influencing some other proteins. Now in this case, we have, when we especially talk about the cytoskeleton, we have the intracellular components. If we say, first and foremost, we have CDC42, we have RAG GTP and ROH GTP. So these are very crucial components when we talk about cytoskeleton. So first and foremost, you can see now this growth factor, as soon as it binds to its receptor, you will see the CDC42 activated. It's in the GTP bound state. Similarly, RAG, it's in the GTP bound state. So is ROH GTP in the GTP bound form. So that means now, so the binding of growth factor to the receptor, it has led to the activation of all of the intracellular components which are involved in regulating the cytoskeletal change. Now similarly, you can see that now the activation of these intracellular components, it leads to the activation of the effector proteins. Like in this case, CDC42, it leads to activation of the VASP, that leads to the activation of ARP2 by 3, and then finally followed by the polymerization of the actin. Similarly, we have RAT GTP. Again, it leads to the activation of an effector protein, that is wave, and finally followed by the actin polymerization. Similarly, we have the Rho GTP, it activates a Rho kinase, and that finally leads to the myosin activity. Similarly, on the another case, it is the Rho GTP, which we have seen in the earlier signaling pathway. So this Rho GTP, what it does, it leads to activation of formin. Earlier, if you say formin, it was inactive, but when signal comes, binds to Rho, leads to its activation, exposes one of the domains, FH2 domains, and then starts the process of formation of the actin filaments. So this is how the actin polymerization is done. And now all of these cytoskeletal changes, they lead to the formation of different components. So especially if we see here in case of CDC42, it leads to formation of philopodia. If we see in case of the RAG GTP bound forms, it leads to the formation of the lamellipodia. And in case of the Rho GTP, it leads to the formation of the stress fibers and contraction. So this was the cytoskeletal elements. Here we focused on the proteins which are involved in the actin cytoskeleton. Next, we will take the microtubules.